cross-section, for example, would look end-on through a cross-section through a nerve bundle, such as a sciatic nerve that might run down your leg. You'll notice that anatomically you can see right away that there are different types of nerve fibers. There are some that are large diameter and wrapped with a thick insulating myelin sheath. And then there are others that are thin in diameter and lack any myelin insulation. And the former of these, the large myelinated cells, are really the ones that the so-called A-alphas and A-betas are devoted primarily to the detection of things that Eric discussed, such as light, touch, vibration, innocuous stimuli, things that we regard as innocuous stimuli, whereas these small diameter nerve fibers, the unmyelinated ones, what we call C-fiber nociceptors, are the ones that really carry out the majority of signaling in regard to how we detect painful stimuli. They're the ones that sense noxious heat, noxious cold, noxious pressure, etc., and then send this information to the spinal cord. So we can see what the sort of just general outlay is of the system. So nociceptions carried out by these unmyelinated C-fibers primarily. Upon injury, they send this information to the region of the spinal cord called the dorsal horn, and then these messages are transduced through a number of synapses to the brain, where we cognitively understand this process as being a pain response. And so, for example, these C-fibers would be the sorts of neurons that were initially activated when Laurie did this vault and injured her knee and felt this acute, intense pain. And the other big breakthrough in this field, as Eric alluded to, was really to understand the molecular devices that make the C-fiber nociceptor what it is. In other words, the proteins that endow these cells with the capacity to detect noxious stimuli and to transmit this information to the spinal cord. And I just wanted to, there are basically two interesting discoveries in this area that are worth highlighting. And one is the discovery of a family of molecules of ion channels that we call trip channels. These are found on a variety of these different C-fiber nociceptors shown here. And the way that these were discovered actually was as targets for a number of irritants that we experience, natural product irritants in our environment, the most familiar of which is capsaicin, which is the pungent ingredient in chili peppers that gives them that hot, burning zing. And it turns out that capsaicin elicits that response by binding to this one ion channel on the surface of nerve fibers called TRPV1. And when it does so, it activates the nerve fiber, initiates an electrical potential change, and we sense this as a burning sensation. This is why we sort of conflate the experience of a hot chili pepper with a hot thermal experience. And
these are all discoveries in the last 4 or 5 years?
Well, in the last 10 to 15 years. And similarly, there's another channel called TRPM8, which is a receptor for a natural product from mint leaves, menthol, which we all experience as a cooling phenomenon. And it turns out that this channel is also activated by cold, which is again why psychophysically we experience a mint leaf as a cold sensation. And there are other such molecules, and all of these play defined roles in allowing subsets of these C-fiber nociceptors to detect environmental stimuli, noxious, such as noxious heat, noxious cold, etc. And then another big discovery that Eric alluded to were these this family of sodium channels. That is, he said sodium channels, voltage-gated sodium channels, are present in every neuron in the body. But C-fibers, actually, a subset of C-fibers express a very particular form of a sodium channel called NaV1.7. This has really come to the fore because of people who, such as the one described in the New York Times, individuals who have mutations in these channels. And these mutations really have very profound effects on the pain transduction experience. So these sodium channels are then responsible for taking the initial activation of the nerve fiber by something like capsaicin or heat and conducting the signal along the axon into the spinal cord. So they allow the electrical current to continue flowing. And mutations in this particular channel have very profound and fascinating effects on pain sensation. In individuals in whom this channel mutations render it non-functional, they cannot experience pain. Interestingly, they can experience innocuous stimuli, such as light touch, but they are incapable of experiencing pain. And as Eric alluded to, you can easily identify such children because they lack the protective function that the pain system normally affords us, and do not know when they have burnt themselves, or they may be running around on an ankle that has broken bones, etc. This has grievous consequences for these kids because they have to be watched very carefully because they don't know when
And just to illustrate how dramatically the field is moving, this is only 6 years old.
That's right.
These were discovered just relatively recently.
And then there's a flip side of such mutations in which people have mutations in the same channel that render the channel hypersensitive. So it's sending signals ordinarily in the absence of any peripheral stimulus like heat or cold, whatever. And in these individuals, they have ongoing chronic pain that's unlinked to any peripheral stimulus, and these patients will come into see their physician sometimes with their hands or feet in buckets of ice water because they feel such extreme burning pain at their extremities. And so these studies really show that these nerve fibers are functionally and molecularly specified to carry out this process of pain sensation, and we're beginning to identify some of the molecules that endow them with these properties.
Alan Bassbaum, tell us how the pain goes from the receptors to the brain.
The key thing is that the information travels from the spinal cord up to the brain and really accesses at least two major areas, because on the one hand you have the experience. Where is the stimulus? How intense is the stimulus? You have a sensory component to it that you can detect. But of course the key thing is that there's an emotional experience that the stimulus generates. And so there are emotional areas of the brain that are activated, and there are sensory discriminative areas. So there's no one pain area of the brain. In that sense, Descartes was wrong. The key thing is that there's a matrix of activity that is turned on in the brain, and that generates the pain experience. But that's really to generate acute pain. What really is special is that in the setting of injury and chronic injury, the nervous system changes. The brain and spinal cord of somebody who's experienced the kind of devastating injury that Laurie had is really an altered nervous system. In that sense, there is a